Welcome back. This is our third session of the day for the ACT program. And I'm very excited to say that we have Sanela Bilic joining us. Hi, Sanela. She's a PharmD and an MBA, and she's currently the CEO and president at Venadro. You can see the name down there, Venadro Drug Development Consulting. She comes from a long career at Novartis, where she served as an expert and director in the oncology clinical pharmacology function. And she holds a postdoctoral in drug development and PK/PD from University at Buffalo, and PharmD, pharmacy degree from Drake University. Sanela, we're very super excited to have you here today. This is great. So please, without due delay, take the lead. Thanks.
Sounds great. Thank you so much. Thanks to everyone for joining. I'm hoping to to really cover clinical pharmacology strategy. And when I talk about clinical pharmacology strategy, it really means looking in deep in preclinical, non-clinical space to clinical space. What we're going to cover today is going to be assessment of the preclinical space, because I believe that's where majority of your projects are, and getting us to that first in man dose, and PK/PD considerations during that space. I will I'll go on to my next slide, which is just covering the agenda. I'll introduce the pharmacokinetics and pharmacodynamics. It's going to be a very short slide on that. Preclinical and non-clinical package to support first-in-human studies will be the follow-on. After that, I'll talk about the differences between small and large molecules when it comes to pharmacokinetics, and really just in general characteristics of the molecules. In addition to that, I'll go over the key PK/PD considerations for large molecules, but I'll contrast it to small molecules, just because I'm aware that some of the researchers here are also working on small molecules, so we can talk about that as well. And then I'll lead into the starting dose approach, and I will focus that on the large molecules, just because large molecule starting dose approach is a little bit more difficult than when we compare it to small molecule approach, which simply utilizes the guidance and MRSD guidance that given by the FDA. So we'll start with definitions, and the first one is pharmacokinetics. I'll call it short PK. It really simply means, I'm sure all of you have covered this in your early PhD workups, and it just additional studies suggest what the body does to the drug. Refers to the rate and extent of drug absorption, distribution, metabolism, and excretion. In other words, ADME. It's described by pharmacokinetic parameters like AUC, which we can call AUC, or we can call it AUC 0 to infinity, AUC 0 to a particular time that we choose due to the particular interest in that exposure. Or simply 0 to tau, which is a dosing regimen that we're giving. In addition to that, we're looking at Cmax and half-life. There are other parameters that are calculated, calculated and presented, and they are clearance and volume of distribution. In addition to that, I'm sure you're familiar with the accumulation ratios. You're likely familiar with MRT, and so on. These parameters are all parameters that are used in some ways as we are characterizing the molecule's pharmacokinetic profile. The pharmacokinetics in general is the study of the relationship between dose and drug concentration. We utilize that drug concentration, we understand the dose, we utilize the drug concentration to have an understanding of what type of a PK profile, what type of a profile the molecule has, and how it translates to whether a molecule has a single compartment pharmacokinetics, two compartment pharmacokinetics, three-compartment pharmacokinetics, whether it has linear or non-linear pharmacokinetics, what type of clearances and where those clearances are associated with. That's all really that kind of goes into the details of pharmacokinetics. Pharmacodynamics is the study of what the drug does to body, and it really refers to the relationship between the drug concentration and drug effect. In addition to the drug concentration and drug effect, really thinking about the downstream effect that we want as part of the mechanism of action, it really also is associated with really drug binding to a particular receptor, which gives us a pharmacodynamic effect by decreasing the availability of the free receptors, or perhaps the availability of the soluble receptor that's floating around. In addition to that, I'll touch upon during the conversation here today the pharmacometrics. And pharmacometrics refers to the use of mathematical models that I just mentioned earlier in the pharmacokinetics section, that are based on pharmacology, physiology, and/or disease to be able to quantitatively analyze the pharmacokinetic and pharmacodynamic data. Any questions on just simple definitions that I just covered? No. All right. Go on to the next one. So I'll jump into the preclinical, non-clinical. Some folks call it preclinical, some folks call it non-clinical. It really doesn't matter to me. It's animal studies, the animal in vitro and in vivo animal studies that we conduct to be able to understand the characteristics of the molecule, to be able to assess characteristics of pharmacokinetics, characteristics of efficacy, and toxicology profiles. And so when we think about the in vitro studies, and this is going to be focusing, in particular on the effect of the large molecule effects. We're really looking at the cytokine release effect change. We're looking at the PBMC assessment. We're looking at the receptor occupancy. We're looking for co-culturing of the cancer cells and T cells. We're looking at the binding affinity between the mouse and human, as well as the humanized molecule, as well as the same for the humanized molecule, where we're looking at the cytokine release, receptor occupancy, cytotoxicity, and co-culture with the tumor cell lines, cross-reactivity, and making sure that our molecule has the appropriate cross-reactivity as well as the binding affinity, and how it differs between mouse, rats, and molgus monkey and human. And then finally, we need to have an understanding of the human tissue binding, and understanding how that tissue binding differs from our tox species. When it comes to in vivo studies with the lead humanized molecule, we either look at it if the mature molecule cross-reacts in the mouse target, we look at it as simple regular xenograft tumor models that allow us to understand efficacy in a xenograft tumor model, because the molecule is able to bind to the mouse target and target on the T cells, if it's a molecule that binds to a T cell target. And with that is able to affect the human xenograft tumor, and so therefore, if the molecule cross-reacts with a similar affinity to a mouse target on the T cells, we can run simple regular xenograft tumor models. If the molecule does not cross-react with mouse target, which is actually the case with majority of the PD-1, PD-L1 molecules that are out there, CTLA included as well. Syngeneic and transgenic tumor models expressing the target on mouse tumor cells, as well as syngeneic tumor models where the actually a murine molecule is used to be able to elicit the effect, where we look at the mouse tumor cells is one of the ways to look at the efficacy and just assessment of the lead. In addition to that, there's also transgenic tumor models where a human human receptor is expressed on particular mouse cells of interest to be able to elicit an effect. Now, when it comes to early development, we need to make sure that that we conduct a dose range in the mouse alone, and in combination perhaps with PD-1, PD-L1, and collect the PK in PK satellite groups to assess the exposure to help with starting dose calculations. Now, the reason behind this is to ensure that that when we are translating the exposures, we are not translating the exposures based on the dose, but we are actually translating the exposure based on the actual exposure observed in the mouse where efficacy is assessed. We need to do this across the cross-reactive species, including, at times, including rat and cyno, to help translate the human efficacious dose. This will inform the next steps. And the next steps are also to be able to conduct the DRF tox studies, non-GLP tox studies, as well as GLP tox studies in cross-reactive species, which can be rat only, cyno only, rat and cyno, or perhaps mouse if rat does not cross-react. Now, in many situations where I've been in and mouse had to be used due to rat not being cross-reactive, I try to, because of those experiences, I try to avoid mouse use due to the animal size and issues with interpretation of toxicity, because of the just sometimes very weird tox findings that are associated with this type of a animal species. And so, if we can run it in cyno, I would rather run it in cyno only and not look for a secondary species. If rat doesn't cross-react in that sense. So the reason why all of this is important is we need to be building appropriate data set that would allow us to ensure that we have adequate amount of PK data to be able to robustly assess how this molecule behaves in vivo in early studies. So, and then usually these studies are in vivo efficacy studies, to be able to then move it on to the next part of the development, where we're looking at translating the efficacy in mouse to be able to inform what doses we're going to be testing in our DRF non-GLP tox studies, and then as we move on, utilizing those DRF studies to be able to inform and conduct the GLP tox studies in cross-reactive species. Now, I talk about the rat only, cyno only, rat and cyno, or perhaps mouse if rat doesn't cross-react. I'm talking about these potential options because of the very recent FDA guidance that suggests that we could perhaps, because of overuse of cynomolgus monkeys, we could perhaps go with a single species, and if your molecule cross-reacts in rat, you can go with single species rat only. If your molecule perhaps, if we discount that particular guidance that just came out, if your molecule doesn't cross-react to anything else, you could also go with in vitro IND only, which requires pretty rigorous in vitro assessments of the molecule to ensure that safety of the molecule is assessed. But why am I covering all of this now? And it is because I think it's important for everyone to be aware that this preclinical or non-clinical development includes workup in a cross-reactive or non-cross-reactive species to assess the PK before we go into the DRF and GLP toxicology studies. In order for us to have appropriate, understanding of what time points to collect. In our GLP tox studies, we need to conduct very robust DRF tox studies or dose range finding studies in animals where we collect the PK, we collect the ADA, we look at the recovery to be able to understand the impact of ADA on PK profile as well as safety. And so when we are looking at the conducting of the GLP tox studies, we're usually looking at multiple dose administrations in the GLP tox study, just like in the DRF tox study. But that study is at that time point very much de-risked in regards to safety part. However, it's also de-risked perhaps if we observed really high immunogenicity and exposure decreasing tremendously in our DRF tox study. One of our options is perhaps to go up in the dose to ensure that that we're going to have adequate exposure over a 4 or 5 week period if this is a 4-week study, or alternatively it will actually help us understand, whether that ADA presence is going to have a detrimental impact on safety of the molecule as well. And so making sure that we're collecting adequate PK and ADA information in the GLP and DRF tox studies, as well as the early PK studies, is really, really important. And it's money well spent to be able to then utilize that data to predict human PK profiles in essentially in humans, in first-in-human studies. When it comes to the collections of the PK data, both in the DRF as well as the GLP tox studies, I usually collect day 1, day 22, and then day 29. Day 1 and day 22, we collect the data in all animals. So essentially all in life as well as the recovery animals. And on day 29, the all animals, quote unquote, we collect data up to 24 hours after which point we see the in-life animals sacrificed. However, the recovery animals stay on beyond that day 29, 24-hour time point, and we usually collect, with a 4-week tox study, we usually collect additional 6 weeks of PK data in addition to the ADA data. So that's really the coverage on the GLP tox study. Are there any questions on the DRF early PK in mice efficacy, DRF or GLP tox study planning, and really in regards to the PK aspects? No. All right. I'll move on to the next section, which is the section on small versus large molecules. In small molecules versus large molecules, it's also separated onto the next level separation, and that is small molecules are small molecules. They range in the level of 300 to 500 daltons. Perhaps there are some small molecules that are a bit larger, perhaps even 1,000 daltons. Some of the antibiotics are as big as 1,000 daltons or one kilodalton, but I still consider it small molecule because it's a chemical entity. Then we have monoclonal antibodies, and I also have a listing here for antibody drug conjugates. I'm not really sure whether there's any workup within your group in regards to the antibody drug conjugates, but I wanted to list it here. So when it comes to small molecules, analytical assays that are used to measure small molecules are usually LC-MS. We usually measure free or bound drug, and in regards to dosing, it's usually oral or parenteral. Our goal is to ensure that we have oral absorption and ability and bioavailability that's adequate to attain exposure needed for efficacy. But in some cases, the IV or subcutaneous administration is more preferred due to issues with oral absorption and bioavailability. When it comes to small molecules, the distribution is very variable. Binding implies distribution, and it's usually very, very wide. It's variable, but much wider when it comes to small molecules compared to the large molecules or monoclonal antibodies, both including the just simple mAbs as well as the ADCs. When it comes to clearance, The small molecule clearance is really associated with hepatic and extrahepatic metabolism, renal and bile elimination, as well as relatively rapid clearance. The half-life for small molecules really ranges from minutes to hours, perhaps 1 to 2 days. The longer half-lives associated with small molecules is very rare. However, it does happen with certain molecules, and therefore their PK is pretty special, because they usually elicit the PK profile that we likely see with large molecules, including monoclonal antibodies, ADCs, and perhaps fusion proteins. When it comes to the pharmacokinetics for small molecules are variable. They're usually independent of pharmacodynamics. They are both linear and nonlinear, and they can be explained via many different compartmental behaviors. 1 compartment, 2 compartment, 3 compartment behaviors. In addition to that, non-linear pharmacokinetics associated with mentioned clearance, but most of them have normal 2 compartment kinetics that are linear. When it comes to monoclonal antibodies, and I'll cover here monoclonal antibodies and antibody drug conjugates, just because they are mostly very similar between each other. The analytical assays that are used to measure are ELISAs, gyros, LC-MS, MS. When it comes to ADCs, it's mostly LC-MS, MS today. However, the ELISAs are still used for early preclinical work. When it comes to the analytes that are measured for monoclonal antibodies, we're measuring total and free IgG and immunogenicity. For ADCs, we're measuring the total IgG, we're measuring the total ADC, we're also measuring the free toxin as well as immunogenicity. When it comes to dosing, because of the low pH instability associated with monoclonal antibodies, including the ADCs, parenteral administration is the only way to administer these molecules. So IV or sub-Q is the approach for dosing administration of the large molecules. Distribution is pretty small, and binding implies clearance. And what I mean with clearance, it's binding to the receptor. Therefore, the drug is no longer in blood, and it appears to have cleared from blood. And it's very similar to the ADCs. When it comes to the clearance of monoclonal antibodies, it's really associated with receptor binding, receptor mediated clearance. There's basal catabolism, essentially breakdown of the protein in lieu of binding to the FcRn. IgG level dependent. Essentially the receptor mediated clearance is IgG level dependent. Renal bile is renal and bile is uncommon, and they are usually explained or described as having relatively slow clearance. When it comes to clearance or really characteristics of monoclonal antibodies and ADCs, what we're going to see is half-lives in mice perhaps that are 1, 2 to maybe 5 days, half-lives in rats that are very similar to that, perhaps up to 8 days, where monkeys actually could have half-lives anywhere from about 7 days to, unless the molecule has a very large target expression, and therefore in a normal monkey. But mostly it's about 7 to about 14 days. I have worked on molecules that have had even longer half-lives in my monkeys, but in general it's between about 7, 8 to about 12 to 14 days as we are looking at GLP tox studies. Now, some of these parameters, in particular half-life, can be quite impacted by target expression that I just mentioned, and they can also be quite impacted by the presence of ADA or anti-drug antibodies or immunogenicity. The ADA will bind up your drug, and it will essentially clear it from the systemic exposure or presence. though it's still there, we just can't capture it because we are measuring it with different types of assays that are requiring the molecule to be free to bind to a particular receptor on the plate. When it comes to overall PK profiles, these molecules, whether they are monoclonal antibodies or ADCs, are described as the two-compartment behavior molecules. They usually have nonlinear PK. However, some molecules, some soluble receptor binding molecules have linear PK. But there's really large variability. In general, every molecule will have nonlinear PK. It's just a matter of how low do you go, because the reason for the nonlinear PK is associated with the level of the target available. And so if the molecule, if the particular target is at a low abundance, you're going to see, you're going to need to look really, really low in regards to the doses to be able to assess the non-linearity or faster clearance associated with that. Or perhaps you can go with a higher dose and just continue looking for it after a single dose, perhaps with some molecules after 3 weeks, with some molecules after 3 months, with some molecules after a year, you will eventually see the nonlinear behavior that's associated with target-mediated disposition and change in clearance due to binding to the target, and essentially that gradient between the PK profile and presence, the concentration of the drug and the concentration of the target available to be able to be bound to.
Sanela, this is Enrique.
Yes.
I have a quick question for you, which is probably, it's going to be probably a long answer, but anyway,
Sure.
the question is, when we look at MABs and ADCs, right, and we're looking for those nice PK, PD behavior charts, right? Is there any considerations? Is there any way that we can get better data and based on IV or SC formulations for parenteral delivery? Does it make a difference? I know that a lot of big pharmaceuticals are moving into subcutaneous, right? And they go with novel formulations for extended release and so on, right? But my question is, we don't need to worry about too many things right now. We just need to worry that we get good data, good charts, right? So that we can articulate the argument that these products look good from the PK/PD perspective. So is there any considerations in terms of formulation for MABs and ADCs, or it doesn't really make a difference? Because I assume that SC is a little bit more pricey and longer to develop than an IV product?
Yes. Yes. So if it really depends where your molecule is going to be developed in. There is a push toward utility of sub-Q just because it allows easier administration. It decreases the amount of time the patient needs to spend in getting into the doctor's office to get the drug administered. With majority of the sub-Q administrations can be administered at home if the molecule is safe enough to be administered at home. So I mean, there are definite benefits associated with utility of sub-Q administration. However, there are costs associated with that. And what my recommendation would be, if your plan is, and if your patient population is if your patient population and the disease are allowing the utility of sub-Q, I would start with making sure that your drug material is concentrated enough, because you need to ensure that, again, sorry, I have to go back to the one more point. It depends on the pharmacokinetics of the molecule and how it relates to the target availability. If your target is it's not a druggable target, right? A sub-Q administration is probably not the way to go, because you're likely going to require really high doses, which translate to really high I mean, it translates to high doses, right? So, with some molecules that I worked on, we needed to go with a dose of, 10 or 15 milligrams per kilogram, okay? Assuming that a human body is 80 kilograms, that translates to, 800 to 1200 milligram dose. It is almost impossible to get the drug concentrated enough beyond 200 milligrams per ml, okay? And so what that means is one sub-Q injection is 2 mLs, and that essentially gives you a dose of 400. Now, the patients will be fine with going with perhaps 2 injections of 400 to get you to a dose of 800. But if you need a dose of 1200 milligrams or 1600 milligrams, for instance, it's kind of impossible. So it's very dependent. All of this planning needs to happen really early on. We need to have a really good understanding of your targets, of the patient population, the availability of the target, availability of the soluble target, in addition to the target on the cells, to be able to make that decision early on. But if, again, going back to that, if your target is highly abundant and you need really high doses to cover it, sub-Q is probably not a way to go, and therefore you stick with IV. Still work on the IV formulation that's also concentrated, because it will help with vials and so on. But that's one thing. Now the other thing, sorry, I'm going into another direction, but your question prompted me to think about this. The other thing that majority of the companies are going in direction of is utilizing flat dosing, so fixed dosing, so milligram dosing instead of milligram per kilogram dosing in the MAB and ADC space, more so MAB space than ADCs. ADCs are still in that milligram per kilogram and BSA dosing, but in particular with MABs, and one of the major reasons why we're going in the direction of the flat dosing is to perhaps be ready for sub-Q dosing, because you have a single injection, you're not having to calculate a dose per patient and so on. You go with a flat dose that allows you to have more of a straightforward switch to sub-Q. But in addition to that, even if you're stuck with IV, going with a flat dose decreases the amount of mistakes that happen in dosing due to the calculation errors and so on. And so that also helps with preparing the vials. So if you know that your dose is going to be, let's just be simple, the dose is going to be 240 milligrams, which is 3 milligrams per kilogram. So it's going to be 240 milligrams, and that's your dose. You're going to be, planning your vials to be 240 milligram doses for everybody. So there is no waste associated with dosing in that way as well, utilizing IV in addition to sub-Q. Sorry, I know it went over what you asked, but hopefully that answered the primary question you had. Okay, great. So now when it comes to pharmacokinetic behavior, as I said, it's two compartment mostly. We do see nonlinear PK. When it comes to ADCs, though, the profiles that we see are very are different, right? So when we are measuring the total IgG, when you compare it to just a naked monoclonal antibody, are very similar. We actually see normal PK profile with that. However, the total ADC, depending on whether this ADC is a regular ADC, like the DM1, DM4 ADC, or even MMAE, utilizing the I'll say utilizing the initial ways of generating ADCs, you will see a faster clearance associated with ADC. You'll see very similar Cmax, but faster clearance, similar clearance for up to like 7 days, and then that clearance changes as the ADC essentially, in a way, falls apart. Okay, it doesn't fully fall apart, but the payloads payloads do go down, and so therefore we're following the free toxin as well to ensure that ADC is truly not fully falling apart when it comes to the free toxin, because we don't want to see high exposure of the free toxin. But I just wanted to make sure that you're aware that it is normal to see total IgG and ADC in a sense diverging from each other because of the characteristics of the ADC, where the free toxin actually does fall off, but at a slower rate than we would than at a slower rate than we would expect to see free toxin administered on its own. Any questions on this part? There's much more coming in regards to the small molecule to large molecule differences. No. Okay. All right. Now, when it comes to the small molecules, preclinical models can be really multiple species. It's not affected by anything. Where monoclonal antibodies and ADCs, it's limited a number of species due to the limited cross-reactivity observed. When it comes to the PK drug-drug interactions, drug-drug interactions are very common, and they're common because they are associated with enzyme induction or inhibition impact. Where when it comes to monoclonal antibodies as well as ADCs, it's really not that prominent as small molecules, but it is still present, and we'll talk about that in a little bit as well. When it comes to ADCs, there is additional risk with ADCs more so than mAbs, just because of the payload and what's observed with the small molecule component of it. And so we think about it a bit more when it comes to both the IND package that we put together, depending on how novel the payload is, or how not novel the payload is. The IND package would look slightly different in regards to that, and in regards to the preparations for understanding the drug-drug interaction potential. When it comes to purity, small molecules are high, have high purity, where monoclonal antibodies as well as ADCs are and may be heterogeneous. Impurities are very well characterized, and they're often related to toxicities. Impurities are very difficult to characterize with large molecules, because their large molecules are, essentially developed via live organisms, and so it's very difficult to characterize. However, depending on which impurity it is, which we are very well aware of many impurities, that may interfere with PK and ability to quantitate the PK, as well as can impact the immunogenicity of the molecule. More impurity there is, perhaps changes in glycosylation will have an impact on immunogenicity of the molecule, as well as toxicity when it comes to antibody drug conjugates. Immunogenicity. Generally, small molecules are not immunogenic. There's, in literature, 1 or 2 molecules that will elicit an immune response, but in general are not immunogenic. When it comes to monoclonal antibodies as well as ADCs, they may be immunogenic. Monoclonal antibodies that are currently developed, in form of checkpoint inhibitors and immune agonists, I'm seeing more and more of these molecules being very immunogenic in preclinical species, where in human that immunogenicity does decrease, but it's still present. And then when it comes to immunogenicity of ADCs, they may be immunogenic just like monoclonal antibodies, but however, there's a higher risk associated with immunogenicity when it comes to ADCs, just because of the toxicity associated with the payload that's associated with it. When it comes to toxicity, it's either target related or non-target related. When it comes to monoclonal antibodies, the toxicity may be exaggerated, prolonged, or delayed, and it is most likely target related, because we are essentially looking for particular target to bind, and if that target downstream effect is relating to toxicity, that's what we are observing. When it comes to antibody drug conjugates, we have on-target and off-target toxicity, and that, that will be exaggerated, prolonged, and delayed. It is associated with delivering of the drug essentially delivering of the payload into the cells that are not carrying your target, or the cells that are carrying your target, but you're not intending for those cells for that molecule to be delivered to that particular cell. And so, for instance, there's ADCs that have pretty significant skin toxicity, and they're actually not developed further. And there's literature information on it, where the reason behind that is that the target was available in the skin, and it's a non-intended toxicity. However, it's an on-target toxicity because it's really important to choose the right target for an ADC to ensure that the target is only present on your tumor cells and not on any normal cells, to decrease the effect of toxicity that's caused by the payload. Any questions on this section? All right. I'll go on to the ADME. And so with ADME, for small molecules, you have the profiles that are associated with intravenous exposure, here in blue, intraperitoneal exposure in red. You would perhaps say that IT and IV administration are very similar in exposure, and I would agree with you. However, for a large molecule, I prefer to see IV administration than IP administration, just because, if a certain vein is nicked during the IP administration, you will see erratic PK profile, and that just makes it a lot harder to predict what we're going to see in patients. And therefore, utility of the IV administration in mice in particular is something that I prefer to see, because it really allows for easy administration, easy prediction of the profiles and exposures needed for efficacy. Again, for the IM administration or even sub-Q administration, we see that essentially a subdued Cmax, but prolonged time after administration. And then with the oral, we don't see that instantaneous Cmax. However, we see a prolonged exposure and a very long absorption period, and then distribution and clearance. When it comes to large molecules and monoclonal antibodies, really that absorption is associated with associated with and also affected by the ability to be absorbed via lymphatic system, via convective transport. When it comes to subcutaneous administration of the molecule, when it comes to just even IV administration, the absorption as well as distribution are highly impacted by the level of the target in the body of the patient, and therefore you will see that in a patient population, and I'll talk about this actually a bit more in the next few slides, but you'll see that a healthy volunteer patient population is going to have much longer residence time than perhaps an oncology patient population that has much more of the target. And so both the absorption as well as the distribution are going to be impacted by the level of the target that's available in body. I wanted to kind of touch upon the sub-Q dosing as well here, just because subcutaneous administration is becoming more and more popular, as Enrique had just mentioned, and so absorption is done through the lymphatic system, as I mentioned. It allows for self-administration. It's often utilized and administered as a pre-filled syringe or auto-injector. There we utilize a dose of, essentially a flat dose to ensure that we develop only a single dose pre-filled syringe, so that manufacturing costs are decreased in regards to that. The doses can be administered at different sites. However, as I was talking about earlier, the doses should not exceed 2 administrations in one day or during the same time, just due to discomfort to patients. And in general, I'm talking about 1 ml administration in general in patients. However, I know that some drugs are administered sub-Q as 2 ml administrations that will increase pain. However, it is able to be administered at 2 mLs, which allows us to have a dose. If that concentration is 150 or more, perhaps 200, it allows a dose of 800. If it's 150, it allows a total dose of 600 milligrams. Again, just to put that in context for you, that's essentially a dose of 10 milligrams per kilogram for 800 and approximately a dose of 7.5 milligrams per kilogram for 600 milligram dose. In general, bioavailability is moderate. It's 40 to 60%. I have worked on a molecule that have had bioavailability of 80%, but in general, on average, it's 40 to 60%. And so there's really not a good animal model to predict the bioavailability in man. And so initial studies in man are usually done the IV, and then they're switched to sub-Q, or if you're certain that your patient population is going to be would benefit more with a sub-Q administration than a home sub-Q, tox program needs to be developed to be able to support an initial sub-Q administration in your first in man studies. Any questions on this? All right.
Yes, Sanela.
Yes.
Can you speak to the chart that you have in the lower right corner?
Yeah, so this really just describes the difference in the PK profile. If you're looking at, so the blue and red are examples of the PK after an IV administration, and the light blue, green, and purple are associated with sub-Q administration. And really, it's not as intuitive because it's drawn on a log, semi-log y-axis, so you can't actually see the true differences between a dose of 10 mg per kg and 20 mg per kg. So there are slight differences, but in reality there are larger differences associated with if we were to plot this out on a linear plot, you would see actual differences that 50 is lower than 150 and 150 is lower than 300 when it comes to sub-Q, and the same goes for the 20 milligram and 10 milligram per kilogram IV. In general, really the point here is that with IV administration, we see really high Cmaxes and, that gradual decrease in concentration, so gradual and slow clearance, where with sub-Q, we see that initial absorption that happens via lymphatic system, and then a slow clearance associated with it. I like to see this with molecules that are cytokine related molecules, if you will, where we try to avoid high Cmaxes and allow for that gradual exposure of the cytokine, and avoid the perhaps increase in IL-6, TNF alpha, interferon gamma, and any of the clinical signs that are usually associated with high concentrations of the cytokine that's administered to treat a disease.
Thanks.
No problem. So the next one is the distribution. So when it comes to small molecules, I covered this earlier as well, but can be large or small, and binding implies distribution. When it comes to monoclonal antibodies, they're small distribution, and binding implies clearance. When it comes to monoclonal antibodies, they exhibit both specific and non-specific binding to receptors, and so in addition to binding to the receptors, we also have binding to the neonatal receptor FcRn, which is found in most tissues, and in my mind, that is a receptor that is utilized to, I call it as a saving mechanism, not a clearance mechanism, but saving mechanism, because if the molecule is involved into the cell, it will be broken down via lysozymes to smaller proteins. When that same molecule is bound to FcRn, the lysozymes are not able to break them down, and therefore the FcRn is a saving mechanism because it will eventually spit the molecule back out to allow for that low, slow clearance associated with the molecule. When it comes to the volume of distribution, steady state volume of distribution is very low when it comes to large molecules. It's usually 3.5 to 7 liters in human. I've seen it as high as perhaps 10 liters in human. It really depends from molecule to molecule. When it comes to prediction of the human PK profiles, I either use a value of 5 liters or a value of 8 liters. So that's just a simple arithmetic associated with predictions of the human Cmax and so on, especially at low doses. When it comes to small molecules, honestly, I've seen volumes of distribution that are anywhere from about 2 liters to 500,000 liters for a small molecule. So there are huge differences when it comes to small molecules and large molecules, as well as within the small molecule space as well. So this is really just the general schematic for antibody disposition, and so really I've covered this with FcRn, where the FcRn is that saving mechanism. The disposition of antibodies is mainly determined by the combination of both specific interactions with the target ligand as well as that Fc framework, is essentially in particular FcRn framework. Is cleared via RES system, including the Kupffer cells, macrophages, and the spleen, and other cells and tissues in the liver. We'll go on to the metabolism. When it comes to metabolism for small molecules, it's primarily hepatic and an extrahepatic metabolism. When it comes to monoclonal antibodies, there's really no metabolism. Metabolism is exchanged or is really associated with catabolism, which is enzymatic degradation of the molecule. And then, as I was mentioning earlier, FcRn saving mechanism, which is described. Oops. Apologize for that, which is described in this graph here, where those molecules that are not bound to FcRn are the molecules that are broken down and are not able to so the molecules that are here free of the FcRn get broken down to smaller proteins and are cleared, where those molecules that were bound to the FcRn are able to be released out into the circulation, which again is the reason why IgGs have such long half-lives compared to small molecules or really any other molecules, any other large molecules, fusion proteins and so on, that don't have the Fc portion that is responsible for binding to the FcRn. Actually, any questions on this metabolism portion? No. All right. So I'll continue on to elimination. When it comes to small molecules, elimination is renal and bile elimination, and it's relatively rapid clearance. I was talking about molecules having half-life in minutes to perhaps in 4 days, where when it comes to monoclonal antibodies, the clearance is associated via receptor-mediated clearance. It's Ig level dependent clearance. It's a renal bile clearance is there, but it's uncommon, and it's mostly associated with broken down proteins. And in general, it's a relatively slow clearance. What I'm showing here is really just pictorial of the cell surface target, where we have the the targeting, the molecule binding to the receptors on the cells, or the soluble target available, available in the blood, and the molecule binds to it. And so that's essentially, the drugs, quote unquote, when it comes to large molecules, get cleared, but that clearance is really associated with binding to that receptor on the cell, or on the soluble target. These are some simple models that are used to describe the monoclonal antibody PK. And so these are traditional PK/PD models that assume that the amount of the drug target complex is negligible relative to the total amount of drug in the body. They're integrated mechanism-based models of the TMDD that incorporate the binding and stoichiometry of the drug target binding. So that's really associated with the soluble target and the FC, the free receptor and the receptor complex. And then, in addition to that, these models may be utilized to infer the time course of the inaccessible system variables, such as in vivo density of the drug target complex, and provide a suitable platform for getting an apparent pharmacodynamic implications of that target-mediated drug disposition. Now, the target-mediated drug disposition is associated with nonlinearity, and changes in the PK profile. In my next few slides here, we're going to talk about the PK associated with nonlinearity, but I'll cover it actually here as well. So you'll see on this slide right here, where lower doses have much faster clearance, and that clearance actually changes. It slows down, until we get up to the high dose. However, the actual clearance goes back to the fast clearance once we essentially reach an inflection point, where there is more target than the drug concentration, and therefore we see much faster clearance. And so I call this initial alpha clearance, and then this is the beta clearance, and then this is the gamma clearance. Majority of the gamma clearance actually equals the alpha clearance that's associated with the initial target-mediated disposition. Any questions on what I just covered? Okay, all right. So review of the monoclonal antibody PK/PD. The key to understanding the PK/PD of biologics is to really understand the target. Target expression, level, and pattern are really important, whether it's a soluble target or cell surface or both. Some molecules bind to targets that are on the cells, but those cellular bound receptors actually slough off, and so they shed. And we have to think about ability to saturate both the receptor on the cells as well as the receptor, that shed receptor in the blood. We need to think about the turnover rate of that target. We need to also think about the difference in expression between disease versus healthy humans, animals, and humans, and then polymorphisms in target, and the impact on ability to bind that target to on particular cells. The PK and PD therapeutic monoclonal antibodies are quite complex. It is dependent on both the structure of the antibody and the specific antigen target. I would say in very broad terms, antibodies that are directed against cell surface antigens often exhibit nonlinear PK profile, while antibodies directed against the soluble receptors often exhibit a linear PK profile. But if you'll remember what I mentioned earlier, really all of these compounds will have nonlinear behavior, due to the fact that we just need to, not due to the fact, but perhaps we just need to follow it long enough or go low enough in the dose to be able to observe the nonlinearity. There are numerous influences that need to be considered: receptor shedding that I just mentioned, the patient disease state, concomitant drugs, and the physiology of the system that's being targeted. The PD of most antibodies appears to best be described using indirect response type models. However, conducting PD evaluations requires a clear understanding of the interaction of the antibodies with the immune systems, and such models will continue to really facilitate the evaluation of the new therapeutic antibody candidates. So that's really when it comes to overall review of the mAb PK/PD, but when it comes to assessment of the mAbs, when do we need to start? I started out the presentation with having assessments really early on to be able to assess this, and so as early as the target selection and its viability to candidate selection, assessment of the early in vitro pharmacology, in vivo pharmacology, non-GLP DRF tox, IND enabling GLP tox, and then beyond in man to be able to file the BLA. So it's, we need to have an understanding of the PK/PD, and PK/PD, can be focused on, even a single concentration. So when we're looking about in vitro pharmacology, obviously in an in vitro system, we're not going to see dynamic change in the concentration, but we can have an understanding of that single concentration or a multitude of concentrations and look for a pharmacology effect via indirect tumor models or indirect cell models and so on. Indirect effect cell models and so on, and then in vivo pharmacology, where here I'm talking about the efficacy studies. I'm talking about the PK studies to be able to understand what was the exposure that was observed in the tumor models that will translate to the efficacy in humans. Any questions on what I just covered here? All right. So when it comes to assessment of the PK profile of molecules, what you're going to usually see in a tox study or in early mouse studies is utility of non-compartmental analysis, or NCA. However, it only describes the tip of the iceberg. So, what I mean with that, it's really just trying to utilize every time point that was measured to give you an approximation of overall exposure, overall parameters like AUC, half-life, Cmax, and so on. When it comes to modeling can provide much more information. It provides the opportunity to model and then simulate different dosing regimens while looking to attain the same exposure, to attain efficacy, and perhaps avoid a toxicity, because we are able to, from the single dose, we're able to simulate what multiple doses would look like in particular species, whether it's mouse, monkey, or human. When it comes to the pharmacokinetic characterization, it really depends on whether the molecule is cross-reactive or non-cross-reactive in particular species where we're looking at. The case in point here is Kadcyla, which was Genentech data published data in their BLA. It is important to choose the right species to be able to assess the PK behavior. It doesn't make sense for us to be predicting the human PK profile based on rat, which is a non-specific binding species. It is more important for us to be predicting it based on cyno data, where we see actually faster clearance associated with the lower dose, because we expect that there is some HER2 present even in a healthy cynomolgus monkey, where we're going to see target mediated disposition of the molecule. When it comes to, again, large molecules in particular, bioanalytical aspects are a bit more difficult to put together as we compare it to small molecules. Knowing what we're measuring is of utmost importance. Many analytes can be measured. We can measure free drug, we can measure total drug, we can measure free target, we can measure total target. So free drug is the drug that doesn't have the target. It's not bound to the receptor. It's not bound to the soluble receptor. It's just free. The total drug is the drug. It allows to understand it. Essentially, it utilizes a step which cleaves off the receptor from drug, and it allows us to understand what the total drug concentration is. Free target is really the target that's perhaps available after the drug has sucked up all of the rest of the target. The total target includes all of the target that's free, as well as the target that's bound to the molecule. Soluble receptors, metabolites, and that's case by case, and this is in particular important when it comes to ADCs, because we have the total antibody and we have the ADC as well as the small molecule free payload. And we need to be looking at the endogenous counterparts, anti-drug antibodies, and perhaps neutralizing antibodies that are present or that get generated due to the administration of the molecule. When it comes to analytes in general, not all analytes need to be measured. So when it comes to non-clinical development, I like to see total antibody done, because honestly it's cheap and it's easy to understand the profile of the molecule. When it comes to clinical, I'm more interested, especially if I know that the molecule is going to have TMDD. I'm way more interested in understanding the free monoclonal antibody concentration than the total, because that will not tell me how the molecule behaves and how much of the target there is and how do we drug that target. And so free monoclonal antibody assay would be of importance when it comes to clinical, because we can then use that in lieu of any PD, because we'll know that once the PK is saturated, then we know that the target is saturated. In this way, free MAB assay also behaves as a PD readout, because if it's cleared faster, we know that the target is also not saturated. When it comes to the total target, I would rather get the total target than the free target, because again, the free target is usually with a molecule, or usually with large doses, you're going to see no free target available, and so majority of your readouts are going to be zero. I kind of like to see the numbers associated with the total target, because it allows us to actually fit that and predict, if we were to dose, right now we're dosing monthly, and if we were to predict what every 2 months would look like, what every 3 months would look like in regards to the target suppression or a binding. So that's kind of where total target is important. Again, there's really not much dynamics that we can do with the free target. Otherwise, we'd have to wait for a very long time for the free target to come back up to be measurable. And then finally, the ADA, and within ADA, we're really talking about the screening, confirmatory, and titering ADA, as well as the neutralizing antibodies. When it comes to the initial, three tier ADA, that is something that I like to see as early as possible. So, we have it for our phase 1 study. Beginning of our phase 1 study. When it comes to neutralizing ADAs, that really, majority of the companies have this developed by phase 2. I like to see it as early as possible, especially with my biotech clients, because of the fact that neutralizing ADAs are pretty detrimental for the program, and so actually having an understanding of neutralizing ADAs as early as kind of like your expansion in a phase 1 trial allows the investors that are interested in your programs to feel a lot more comfortable with the decision to buy the molecule, because of perhaps lack of neutralizing ADAs. Now that could be opposite, right? The presence of neutralizing ADAs can be detrimental for the program and decrease their interest. But it's so it's a decision to be made. Again, I like to see it. I like to see the NDAs present, NDAs measured, and as that really does help with decision making. When it comes to large molecules, immunoassays are most common to measure it. However, LC-MS is more and more used, in particular with ADCs or it has been used with large molecules when immunoassays are not able to be done in timely manner. With LC-MS/MS, we're specifically looking at particular part of the protein that's on the molecule, where with immunoassays, we actually need to develop very, very specific reagents that require immunization of animals to create either monoclonal or polyclonal antibody that would be very specific to characterization and binding of binding to our molecule as an anti-human IgG, and so for the reason of timing, LC-MS/MS has been used quite a bit with large molecules, just due to the timing issues associated with the development of reagents needed for immunoassays. However, immunoassays are a lot more sensitive, and therefore that allows us to have a better understanding of the PK profile than if we were to use LC-MS/MS. When we were thinking about the lower limit of quantification for assays, immunoassays can really go down to low picogram per ml concentrations, where LC-MS is probably going to be at low low nanograms to microgram concentration levels for LLOQ. So it's essentially a log difference in the lower limited quantification, which can have an impact on our ability to characterize the molecule. When it comes to receptor occupancy assays, they're often used to measure target engagement on cell surface targets, and this is quite important when it comes to molecules that, as I said, have cell surface targets. However, we can use the free MAB PK assay to be able to discern the target occupancy as well. But having the receptor occupancy in addition to the free MAB assay for PK is a definite plus, because then we can have a better understanding of how the molecule actually behaves, and not just simply predicting it based on PK. Any questions on the bioanalytical aspects? Okay. All right, so PK of IgG MABs, I need to cover. I talked about target-mediated drug disposition a bit, but I wanted to make sure that I cover it here. Most commonly assumed to be occurring with cell surface targets. So, however, it can occur with soluble targets and due to off-target binding. The off-target binding is not very common. However, it can be there. TMDD for cell surface targets often occurs between 10 and 20 micrograms per ml. And so if you remember a few slides back, I talked about that inflection point. And so the inflection point for majority of the cell surface targets actually happens between 10 and 20 micrograms per ml. With some molecules, it goes down as low as 1 microgram per ml, and then with some molecules it goes as high as 100 micrograms per ml, and that really depends on the level of the target. Higher the level of the target, higher the threshold point, and lower the level of the target, lower the threshold point. Whether the TMD is observed or not depends on the amount of the drug relative to the target, the drug dose, the target baseline expression and turnover rate, time points at which samples are collected. So now, for instance, if we are dosing every 3 weeks and we are at a saturating dose over that 3-week period, we will never see, for a particular molecule, we'll never see TMDD. However, if we were to see a patient not get the week 3 dose, and we continue following the PK profile, we would actually see the change in the PK profile, change in the clearance associated with the molecule. Sometimes accelerated clearance due to ADA may be misinterpreted as target-mediated disposition, and one of the major reasons why assessing ADA in the early development, essentially phase 1, first-in-man trials, is important to decrease the misinterpretation of ADA with TMDD that's observed. And then just in general, it's dose dependent. Lower the concentration and dose, TMDD is predominant, so we see faster monoclonal antibody clearance. Higher the dose and concentration, TMDD can be saturated. FcRn mediated clearance is predominant, and then we see slow monoclonal antibody clearance. Any questions on this? Now I just want to make sure, how am I doing on time, actually? Sarah or Enrique?
You got like 15 more minutes.
15 more minutes. Okay, so I have
15, 1-5. Yeah.
Okay, all right. So I have like 17 more slides, and that's just not enough to cover everything. So I'm gonna rush to make sure that I cover everything. So the TMDD for soluble target, this is in particular, observed and differences are observed actually when it comes to studying it in healthy volunteers and studying in patients. We're going to see more target mediated drug disposition or nonlinearity associated with patients than with healthy volunteers, just because there's an expectation of a greater target presence in a patient population compared to healthy volunteer patient population. And so what you're going to see on the left side with the IV administration, you see the faster clearance with dotted lines, which is the patients. And then on the right side, with the sub-Q administration, the healthy volunteers are light gray, and the drug A patients and actual patients are in black. And so you'll see that their exposure is lower than in healthy volunteers. And the main reason behind that is the difference in target expression in healthy volunteers compared to patients. When it comes to TMDD for the cell surface target, I talked about that inflection point. You'll see on the left side, the dose-dependent MAB PK TMDD is really associated with you see the very fast clearance associated with very low dose, and you see essentially saturation of the target for the first I'll say 28 days. For the top dose, and then by the time we reach the target mediated disposition, we see the inflection point, and that inflection point for this particular molecule happened around 10 micrograms per ml. And as you look at the PK on the PD on the left side, it's really the target suppression. But as you increase the dose, you increase the duration of the effect. Essentially, we increase the saturation of the target or suppression of the target at 100%. This is essentially from a paper for a CD11 molecule. I can send the paper out if you're interested. If the MAB can be administered at doses and frequencies that maintain trough concentrations that are above TMDD, then TMDD will not be observed. So, with this particular molecule, it ended up being administered on a timed dosing regimen to ensure that the TMDD was taken care of. This molecule is an example of a molecule that had it's actually a cell receptor binder as well as a soluble receptor binder. There was a ton of shedding that was happening, and the example here are actually two patients receiving exactly the same dose. The molecule was measured via a PK assay that was free. So only the drug that was not bound to the receptor was measurable. And so what we saw here was that this patient, patient 101-11101, had very, very fast clearance. Again, at the same dose, both of the patients received a dose of 3 mg per kg. the first patient had tremendous amount of tumor on their PET scans, on their FDG PET, where the second patient, the 201 patient, had very little tumor present. I mean, both of the patients were S9 patients, and both of them were part of this trial as a phase 1 trial. But the sheer differences in exposure and were observed here because of the fact that there was just not enough drug. Even though both patients received the same dose, there was not enough drugs to cover all the target for patient 1 compared to the patient 2. So I'll touch upon this as well, and that's the potential for DDIs in biologics is very low. However, biologics are not free of DDIs. The paper, the figure here on the right is from a Harvey et al. Paper that essentially suggests that that increased target expression leads to increased clearance of the free mAb. Upregulation of the target expression by co-medication can lead to increased clearance of the free mAb. And so if we have a small molecule that has an impact on increasing the target expression, the large molecule PK profile will be impacted with that. And so we really need to be cognizant of the small molecule, kind of downstream effect on the target, to ensure that we're not going to see the potential drug-drug interaction with this, because we will require more drug on board to drug that, to drug the additional target present after the increase in expression. When it comes to expression of some cytochrome P450 enzymes, they can be downregulated by certain cytokines. In particular, cytochrome P450 3A4 will be downregulated by IL-6, and so we work with. I work with a ton of molecules, ton of immune check inhibitors, as well as cytokines and fusion proteins that have an impact on both IL-6 and TNF alpha, as well as interferon gamma, as they have an effect on immune cells. And so the actual drug-drug interaction is the disease drug-drug interaction, where with the changes in inflammation, changes on the cytochrome P450, and changes because of increase in cytokines, we are essentially, with the disease as well as the drug's action on the cytokines, we will see an effect of large molecules on perhaps some of the 3A4 metabolized molecules. And so I want to make sure that you think about, as you prescribe perhaps, or as you think about prescribing or taking the medication, or combining it with molecules, some concomitant molecules that have narrow therapeutic indices, we need to think about their metabolism as a small molecule due to the potential impact on cytokines by large molecules. I'll just actually mention the names. The tocilizumab is the IL-6R IgG antibody, and within their label, they were in vivo studies done for omeprazole and so on. Anti-CTLA-4 and anti-PD-1 IgG antibodies increase production of the TNF alpha, IL-6, and interferon gamma by immune cells, which then have an impact on the cytochrome P450 function. And so these are just biologics examples where actual drug-drug interactions could be impacted. This one is really an example of where the regulatory expectations were changed with this particular molecule, where the FDA is now asking sponsors to describe the effect of the disease on the metabolism of concomitant meds, and hence potential changes on the metabolism of drugs as consequence of curing the disease. And so it's just an example of the correspondence with the FDA for this particular molecule. All right, so I am not going to, in 5 minutes, I will not give it, I will not give it the full attention, but I wanted to start with the starting dose approach, and that is that it really, I like to use this quote because it's a Paracelsus quote. He was alive from 1493 to 1541. And the quote says, I'm not going to read it in German because I can't, but in English, it's all things are poison and nothing is without poison. Only the dose makes a thing be poison. And so, when it comes to starting dose, I often think about of what happened with TeGenero. Okay, when it comes to the large molecules, there's examples of TeGenero with small molecules as well, but I'll focus on large molecules first. 500 years later, guidance for industry and reviewers came out in regards to estimating maximum safe starting dose in initial clinical trials in adult healthy volunteers. So the prediction of the first in human dose for a monoclonal antibody is simple, but yet very complicated. And it became very complicated after the TeGenero case. So we not only need to predict the PK, we also need to predict the PD, but prediction of the PD itself is very complicated because in vitro and in vivo pharmacology data are not always exactly what we expect to see in humans. And so that actual translation is the key here. Having an assessment of very early preclinical models, including the in vitro models, utilizing in vitro models that translate to the human setting is really, really important to ensure that we avoid, and I'm just going to jump to that we avoid the TeGenero incident that happened in 2006, where 6 of first in human study in healthy volunteers, 6 of active patients, 2 placebos, received a dose of 0.1 milligram per kilogram. The dose was administered over 3 to 6 minutes as IV administration. Interesting piece of information here is that monkeys actually received an infusion of 30 minutes. But in humans, it was administered over 3 to 6 minute IV infusion. The volunteers were dosed in 10-minute intervals. The first infusion was started at 8 a.m. The last at 9:10. Within 1.5 hours, all 6 volunteers had systemic inflammatory response. Within 16 hours, by midnight, all 6 volunteers were admitted to ICU due to cytokine storm, multiple organ failure, angioedema. Elephant man is the man in top right. Coma, destruction of the patient immune system, and so on. The problem associated with this was that the molecule was tested in tox study. The top dose was 50 milligrams per kilogram. The top dose was very safe in monkeys, and therefore that was called an NOAEL. When we translated that NOAEL dose to a human equivalent dose, that was at 16 milligrams per kilogram. That selected HED dose was most appropriate, utilizing the most appropriate species was 16 milligrams per kilogram. They applied a 10-fold safety factor, which allowed them to start at 1.6 milligram per kilogram. They also added an additional safety factor by 16-fold, and said that okay, now we're definitely going to be safe at a starting dose of 0.1 milligram per kilogram. They did not adjust the MRSD dose based on the pharmacologically active dose. The fact is the dose of 0.1 milligram per kilogram actually translated to 90% receptor occupancy for an agonist. Majority of the agonists actually have full effect while having 5 to 10% receptor occupancy. And therefore, the TeGenero group did not do the step 5, which adjusted the MRSD dose to the pharmacologically active dose, and essentially led to the development of the starting dose of 0.1 milligram per kilogram, and not where we perhaps needed to be for an agonist antibody. This situation, this incident, terrible incident, was the reason why we have minimally anticipated biological effect level dosing in monoclonal antibodies, and guidance for calculating the starting dose. So we're on time. I have 5 more slides to go. I'm so sorry for taking too long, but I'm happy to cover it if anyone wants to stay extra.
If anybody has any questions and or is able to stay a couple of extra minutes, then feel free to go ahead and ask them. Many of you are signed up for office hours this Friday as well. And if you didn't get a chance to do that, let us know. We can always set up some additional time. So with that, does anybody have anything that is a burning question after all this information? Okay. I think we're hitting our third session of the day slump, Sanella, but this has been, it's a really, really fantastic presentation. You covered a lot of information, and if everybody, gains one thing from today, I hope what they got is this is pretty complicated and it's pretty pivotal. And I think I'm pretty safe to say, right, Sanella, it's never too soon to start thinking about how this is going to impact your drug development.
100%.
All right. Okay.
Is there a specific formula we can use for mouse to human conversion of small molecules?
Not really. I usually use mouse, rat, and dog to do multi-species allometric scaling to get a human PK. Prediction of the human PK based on small molecule data in preclinical species is not as right on as it is for large molecules. There's a lot of issues associated with or a lot of different assumptions that we need to take into consideration, including the metabolism that is quite different and can be quite different from different species. And so it's not as easy as with large molecules for sure. But I can, if we have time one-on-one time, I'm happy to kind of go into more details of that and how multi-species is used to help us get there.
Thank you.
All right. I think this is it then. Thank you, Sanella.
Thank you so much. Thank you so much. And I'm so sorry for running over.
Good.
No, you're great. Thank you so much for the amazing presentation.
Thank you. Have a great day, everyone.
Bye.
Bye.